Yay. So are you able to see my screen now? Perfect. Perfect. And also, I have the chat open here, and teaching during the pandemic helped me how to multitask, so I can talk. And if you don't want to interrupt me, but you want to ask a question and you will forget it later because, yeah, our memories have been short, you can type in on the chat and I will see it right away. So feel free to interrupt me. I will stop and I will talk about your questions. So feel free to jump in at any time. So I want to talk about the specimens. I want to talk about research involving specimens because I've been doing a lot of this. Alex already gave a little bit of an introduction about what I do for my research. And everything that I do, that we do in the lab, involves specimens. And I will tell you why I came to be this biased. And I will give you a little bit of a walkthrough in my shoes so you understand where I'm coming from. I want to start by thanking a lot of people. We usually finish by acknowledging and thanking people, but I want to start with, because nothing that I've done would have been possible without the people in these institutions, without the support of these funding agencies. And I am just so very thankful to many people, all of my colleagues at the museum here, all of my colleagues at Guelph, Alex, Andrew, everybody, and nothing would have been possible without them. I will not name everybody because otherwise I will spend the entire day on this slide, but I just want to start by acknowledging that science doesn't happen in a vacuum. There's no such thing as a genius person that has a bunch of eureka moments. We all need support networks. So I am here because I am a product of the support that I got. If you don't know me, I'm Brazilian. My accent is very weird because I learned German before I learned English. So like my accent is screwed up. And growing up in Brazil has given me a lot of ways in which I'm biased and shape my perspective for the good or for the bad. We all have that. But being in Brazil spurred my interest in nature. I always knew I wanted to be a biologist, always, ever since I can remember. But I didn't know that I wanted to be an entomologist. It was only when I started college that I figured out that insects are the best. And I want to repeat this throughout the class, but I think that flies are the best insects. I do like Hymenoptera. They are my second favorite, but they are second. And I'll tell you why flies are so cool. And I'm hopeful that I will convince you towards the end that flies are really cool. So what I like doing throughout my life and for the rest of my life is understanding insect complexity on all levels. And from molecules to phenotypes, from ecology to genomics, I just love everything about insects. But I am mostly focused on questions, macroevolution, sort of the bigger picture, and taxonomy and systematics, because I care about each individual species. And I'll circle back to this in a little bit. As a person from Brazil, I also care and I think a lot about accessibility. And I saw how my access to things shifted, changed, expanded once I started my PhD in the USA. I mean, it was just a complete change of scenery in so many things, not only because of the language differences, but everything is different. And as a scientist, I care about access to data, resources, information, infrastructure, and support that I mentioned. Brazil's situation mirrors that of many places around the world. Places with substantive vegetation cover also have concentrated poverty and income inequalities. This is a very interesting study that came out in 2019 that shows that 7% of the total municipalities in Brazil harbor about 40% of the vegetation cover, and of those places, 30% they represent 13% of Brazil with economic needs, which there's a parallel between having lots of forest and biodiversity and being a hotspot with lacking resources. And this is true for many places around the globe, especially in the global south. where colonialism and extractivism shaped current economic realities. So this plays a lot about what I think of science these days, what I want to do as a scientist, and why I care about accessibility and how that has shaped my research interest and practice. I'll circle back to that in a little bit. I am also the director of the Lyman Entomological Museum at McGill, and I am an assistant professor in the Department of Natural Resource Sciences. As a Brazilian, I must say I love spring and summer and fall, but the winter still is an acquired taste to me, especially towards December when the days start getting short. Oh my goodness. I also oversee this collection, which is one of the largest in Canada. And it's a very important collection for multiple groups of insects, including Canadian butterflies and Orthoptera. The Lyman actually is one of the best and most representative collections of Orthoptera from all around the world in Canada. And I am also sort of a disclaimer too. I'm also a taxonomist by training. I started off as a taxonomist. I started off describing species using morphology, like poring over microscopes and making lots of drawings. So I have been recently exploring different scientific avenues. As I will show you in a moment, my perspective still pretty much is that of a taxonomist and museum director. In this perspective is that each specimen has value to me. I oversee this collection. I care about leaks. We are in a basement, and who would have thought that putting a museum in a basement was not a good idea? I mean, yeah, I would have if anybody had asked. So I see each specimen as my responsibility, not only because I don't want to be eaten by dermestids, but I don't want to be wet. So I care about each specimen, and that shaped the way I see science coming down the road. And I'm going to talk about that, especially when we talk about genomics. So I mentioned a little bit about my research, but I do have in the lab sort of two foci, focuses. I don't know what the plural foci is, but two different focuses. I care about the organisms themselves on one end and sort of about biodiversity science and community engagement and taxonomy, but I also care about placing them into an evolutionary context, including insect macroevolution of all kinds. I mean, I have all kinds of students working on all kinds of insects in the lab these days, not just flies. I don't force anybody to study flies because otherwise I wouldn't have no students. But also phylogenomics and bioinformatics. And I will explain a little bit about them, what I've been doing on those. And central to everything I do, from taxonomy to genomics to bioinformatics, are phylogenies. I hope you like phylogenies because they're everywhere in this talk. And I see things through a phylogenetic lens. The world to me is kind of just relationships. And to me, phylogenies are wonderful tools, if not unnecessary, a prerequisite for biodiversity research. They offer a window to the past, but also a means for us to interpret the present. And most of my research these days involves phylogeny. And I am very interested in figuring out how we can make more accurate phylogenies, in estimating phylogenies for the groups that I care, and expand our access to phylogenetic tools, circling back to accessibility that I mentioned. And here are just 5 things that we can do with phylogenies. We can understand all these things. And I don't know if you knew that, but phylogenies play a central role in epidemiology as well. The flu shot is made with phylogenies ahead, phylogenies of strains. Maybe COVID vaccines will be made that way one day. Who knows? But phylogenies are everywhere, whether we like it or see it or not. And I have been investigating the microevolution of insects for a while now. And I have worked on many things, from flies to wasps to neuropterans. And I used, as Alex said, multiple lines of evidence, from behavior to morphology to genomics to any fossils, anything else, paleontological, geological data, geographical data. And I love mixing things up and coming up with new ways of harnessing data, especially data in natural history museums. In leading these projects or being part of a collaboration on these projects provided me with an important lesson on reproducibility in science, and especially in terms of vouchering. I'll circle back to that in a little bit. But I want to stick to data availability, access, and reproducibility in terms of phylogenetics. A survey of data availability in terms of the trees themselves in the alignments, the DNA alignments or protein alignments or whatever you want to do it. Here's an assessment of the availability of the data used in phylogenetic studies over time. And if you notice on the, look at the pie chart. I don't have a pointer. I wish I had like a little, I don't know how to do that. I'm not that smart to figure it out. But here, oh yeah, I have one. Like I can do it on my own screen. I took it home after playing with the cat. But 25% of studies only have the data available, either one or the other or both. Just a quarter. So if you need data in term or the results from those studies, you have to beg them, the authors. Once you bag them, then accessibility or data availability increases to 52%, which is better, but still is just a little more than half. And about 33% of data is unavailable. The authors don't respond or the data is not posted online, which is problematic. No matter how much you bag, you don't get that data. And things have been getting better over time. What it says that policy, those are not actually policy, but those were campaigns by the scientific community around the early 2010s to change this. And he has made an effort. But if you look on the gray portion, it's still big. There's still a law. And this is problematic for people that are in the global south, for example, that don't have access to the genetic data, for example, to make the phylogenies. What if what they need is the phylogeny as a tool and they don't have access to it because the authors don't post it online or they don't respond to emails? This is a huge problem. And yeah, I mean, it's something that has happened to us. I mean, if I ask around, I'm pretty sure you have come through this. You have reached that roadblock. Doing meta-analysis must be a nightmare, not only because there are many studies around, but because the data might not be available. But hey, I am no saint here. I have done this. And here's an example. I am not preaching. I'm pointing out the problem, but I'm not being a hypocrite. I am guilty. I was part of this phylogenetic study on owl flies and ant lions. And I'm going to just segue a little bit to talk about this project. One of the most interesting results that we got was that owl flies are actually derived in lions. They're nested deep inside in lions. So they're just derived, glorified in lions, just like birds are glorified dinosaurs. And this project actually has been a little controversial, not controversial, but it has been, I mean, it makes sense morphologically, but people do care a lot about owl flies. And I've been to meetings, that happened to me twice, not once, just twice, that I came to talk to people. Oh my God, you're Gillung, you sank my favorite family. There's no Ascalapri anymore, jokingly. So I love this project, but hey, I put on the alignment and I forgot to put on the tree available. So just a couple weeks ago, I got an email from one of the collaborators, the first author, asking me for the data because I forgot to put it online. So I am on the guilty part where I put just one part, but not all of it. So people have to beg me. I am basically a gatekeeper. People don't have access to it, if not through me. I will fix this, I promise. I don't know how yet, but I will fix this. So this very fact got me thinking, and I had this presentation ready about weeks ago. And when this happened, I changed it. So like this entire presentation is like 80% new because I've been thinking a lot about these things. And this changed a little bit of my current focus and interpretation of what I do as a scientist. How can people replicate our results or reuse our data if we don't make it available? How can colleagues assess our specimen identifications, for example, if we grind them whole for DNA extraction? And again, I've done both things. I'm no saint here. The solution to the problem of data availability is very simple. You just post everything online. When you publish your data, your results, you publish your data. The solution to the vouchering thing is far more complex, and I'll circle back to that. And there's no easy solution for this. So yeah, we need to publish things. We need to do better as a community. And I have always been interested in specimens themselves. And I have always been in and around natural collections. And it's just I'm so very lucky to be able to work at the Lyman Museum. And this photo of me here, the one that was on the flyer, and it's like, I think is the only, we never like our photos. This is the only photo of myself that I like. It has been overshared, so I need to use another one, but like, it's the only photo of me that I ever liked. It was taken at the Bohart Museum, one of the largest in the US, where I did my PhD. I love museums. And naming and describing new species has given me an appreciation for the value of each individual specimen. This is very clear when we work on taxonomy itself, right? We have to pick one specimen to be the holotype and then several other ones to be paratypes. And I have worked on taxonomy for a while now. I love it. I joke all the time that I'm doing the fancy genomic stuff just so I can get tenure and then go back to describing species. I really want that. And I want to talk about funding and in these issues in a little bit too. So here are photos of some of the flies that I described myself or in collaboration. And I work mostly on parasitoid flies. And parasitoids are organisms that have sort of an intermediate lifestyle between predators and parasites. They're neither, but they're kind of both. They're fascinating to me. And I've had the privilege to describe 33 species, either myself or in collaboration. And here, just the flies, because they're the best insects. And I mean, the importance of specimens, each individual specimen for taxonomy and natural history became very clear once I have the responsibility over the Lyman collection here. And I'm pointing because the collection is that way. You can see it, but it's that way. So now I see each specimen not just as a physical object, but as dynamic suites of interconnected resources and data. I see them as magical things. And I greatly appreciate the concept of the extended specimen. This is just a fascinating way of seeing each specimen. An extended specimen begins with the specimen themselves, the physical specimen, the little one in the pen, and then he aggregates data, additional data, all kinds of other data to that one particular specimen, including DNA sequences. Ultimately, an extended specimen can become part of a network of other extended specimens. And you can actually think of a phylogenetic study, for example, of phylogeny, as an amalgamation of different extended specimens because we are linking them through their DNA. I find this magical. But if each specimen is as valuable as I say, why are we not processing, labeling, pinning, IDing, and databasing them all? I wonder this. And here's a little bit of why I think that's not the case. In many cases, it is easier and more feasible to get funding for new collecting trips than it is to fund the processing of specimens we already have. I've come through this. I've tried both ways, and I get money to go collecting more specimens and add to the backlog. But it's very hard to process. And this is especially true for biodiverse and undersampled places. You say you're going to go to Brunel, we'll give you money because we haven't been collecting there a lot. And it is also easier, and that has been my experience, it's easier to get funding to buy fancy toys, fancy equipment, fancy instrumentation. That's true in Canada. Than it is to actually get the funding to run those toys or to hire people to help you run those toys. this is an incredible conundrum to me, because I can buy all the fancy toys I want, and then they sit there because I don't have money to buy the reagents. And now after COVID, everything is more expensive. Try setting up a new lab post-pandemic where tips are just so expensive. And also this focus, and that's very true for Quebec, there's this focus on innovation and novelty at the expense of continuity. But what is novelty and innovation for taxonomy? A centuries-old science that's vital for everything. So this mindset, agenda, whatever, affects taxonomy and biodiversity monitoring and research in big ways. And there is an enormous backlog of specimens everywhere. I mean, that's just the nightmare of every person that works on insect. There's always jars and jars and jars and jars and jars of Malay strep. And in Brazil, did you know that in Brazil, there's a fungus that grows on the alcohol feeding on the specimens? I've pulled out, and I've only seen that in the tropics. I've seen it in Argentina and in Brazil, not here, but they're like fungus specialized on ethanol preserved specimens from Malay streps. So we can, this innovation where I'm gonna collect on this place that has never been collected, plus novelty of it all, we can point the fingers at that. But also, we also have an overwhelming volume of specimens, whether we collect new specimens or not. And there's lack of resources for processing them, for, oh yeah, even above 70%. I mean, yeah, those fungi are incredible. So there's lack of resources, supplies, and storage capability, as lack of funding to hire expert people themselves. And some new approaches have been proposed to speed up taxonomy, to speed up biodiversity discovery, to speed up processing, and those are very welcome. And I believe as long as those efforts are accessible and reproducible, yes, let's do them. We are losing biodiversity. We need to speed up taxonomy. And to me, the main problem don't lie on unconventional approaches to taxonomy, but on the fact that funding agencies, or the way the world is these days, there's an agenda that doesn't quite include the practice of taxonomy and the training of next generation of taxonomists. There's not a lot of support for that. So yeah, I love that comment. I use a centrifuge in a novel or innovative way. Yeah, yeah. I mean, that has been the trick to get some of the funding that I got. I mean, that's incredible. But like this need for innovation and novelties kind of, I like continuity. Anyway, this might be a side rant. We can talk a little bit about that towards the end. And in terms of accessibility, in terms of being Brazilian, of global South and everything, we are in a world dominated by digital data. And I believe it is also urgent that we think about what we do with those specimens, that we find resources and ways to support the processing, that we discuss phylogenetic data, accessibility and all. But I think it's also important that we engage in conversations about how we can best enhance, promote, democratize, and modernize natural history collections in the massive amounts of data they contain. I look at the museum and I keep thinking, oh my God, what people in Brazil could do if only they had access to these things, whether it would be digitally or not. Digital specimens ensure the long-term accessibility of data, not only for current methods that we do, but also for innovative techniques such as museomics and digital vouchers for metagenomics projects, which are very common these days. And I want to bring our attention to this map here. This map indicates all museums with biodiversity relevance around the world. And there's a lot in the global South, but if you look, the concentration still is in the global North. And the funny thing is that Australia and New Zealand count as global North. So even though they're geographically South, they count over there. So they don't think that that belongs with the global South. There's this disconnect between where the specimens are from, because most specimens come from the global South, and where the specimens end up in terms of depositories and who has access to them. Specimens in some of the largest natural history collections in the world may not be accessible to the communities in the global south. Therefore, digitizing specimen data not only increases equity and inclusion in biodiversity research, but it also reduces the likelihood of helicopter science. And someone mentioned here, backup for floods and fires. Oh my God, we had like 3-4 different floods the past couple years here at the museum, a couple fires, resident chipmunks. We had like all kinds of plagues over here. So yes, digitizing data protects data, makes it available, makes it protected, makes it equitable. And not only that, I mean, this is already good enough, but if you think of the pandemic, here's the news from the Natural History Museum in the American Museum of Natural History in Washington, D.C. They basically fired lots of their employees, their staff, because of budget cuts. If one of the largest museums in the world was in financial trouble, imagine the rest. The current global pandemic has exposed not only the precarious existence of most natural history museums, but also resulted in a significant slowdown or stoppage of biodiversity, natural history, specimen-based research at all. For instance, my university and consequently the Lyman Museum, we were closed for most of 2020 and 2021. And now we have backlog of requests that we don't have a lot of stuff to do. So not only we need to start thinking about how to make sure that we make this data available and we make biodiversity research, specimen-based research resilient in the face of uncertainty. And museums in the modern era must emphasize not only staying alive, but also growth toward equity and diversity in visitor populations, access to their collections, and outreach efforts. Who is this data for? Who does it serve? Given this, data from extended specimens, from digitized specimens, provide ways of maintaining research activity during disruptions. And I found this paper that talks about museum business models, and I applied to biodiversity natural history collections too. And I never thought about it this way, but now that I am the director, it makes sense. We talked about clients as opposed to students and all, but like it boils down to money. Sometimes speaking the language, not treating, not accepting, but speaking the language. So here it is. Recent research on museum business models has found that investments in digitization offer museums new opportunities to diversify and expand their funding and income. It has been demonstrated that when most of a museum is digitized, more people find it useful, visible, and it attracts more money. Basically, money makes money. So, am I a hypocrite here? A little bit. I am in terms of data availability and I am in terms of data digitization. But I'll claim I just started. We're working towards thinking and doing more about this. Almost 10% of the museum, about 300,000 specimens from our museum has already been digitized and it's available on Canadese. It's freely available. You can check everything. That's mostly flies, not because of me. I have nothing to do with it, but because the Lyman Museum has a very long tradition of dipterological research, of which I approve. But now we want to expand and make it more available for everybody. And using, so here comes, and let me see what I'm doing in time. I do have 10-15 minutes. Yeah, okay. I am gonna go a little quicker on this part here because I wanted to spend my time explaining how I think these musings, generating this discussion. And now I'm gonna get into this part where I present what we've been up to in terms of all of these things, how this all tie in together with the research that we do at the museum. Using this amazing collection of flies or insects, and then flies, which I have a preference for. We have been working hard on many different projects, from parasitoid lifestyles and macroevolution to agriculture. And I'll talk a little bit about that at the end. One of my current projects aims to understand the macroevolution and macroecology of parasitoid flies. Just briefly, they are organisms that live on or in a host, kill the host, and then complete development. And as you can see, they're very cute. They are so adorable. I really like parasitoid flies. In my opinion, they're some of the most remarkable organisms on Earth. And I think parasitoid flies are more interesting than parasitoid Hymenoptera for one reason. They reinvented the wheel over and over and over and over again. Some estimates say that parasitoid flies evolved this lifestyle, which is very complex, about 20 times. And some estimates say 100. We don't have a comprehensive tree of life for flies yet, but that's a lot compared to one or twice, depending on the hypothesis for Hymenoptera. They reinvented the wheel maybe once. Flies all over. And this research wants to understand why this happened, not only why parasitoid flies evolved over and over again, but wants to understand why flies are so not specific. Here you can see a comparison of parasitoid wasps and parasitoid flies in terms of how they relate to their and I'm laughing because I'm reading the comments on the chat and I'm laughing so hard. We're all so polarized over flies and wasps, and I love that. I mean, if that's what we disagree on, we are such a good community. This is amazing. So flies, they tend to be generalists, both in terms of the total number of hosts, 5 phyla versus 1 phylum in Hymenoptera, but also individual species tend to have a breadth of hosts. And I have a hypothesis for that. I think it's because they breathe differently. And nobody has looked into that yet. I haven't had time to do it, but I have this hypothesis that the way the flies breathe inside their host, they make a little funnel, a little straw called a siphon, or they make the host do it for them when they're lazy. And then they breathe outside air. That frees them up from selection against suffocation, meaning they are more free to invade new hosts because they don't have to be so in tune with the immune system of the host, as opposed to Hymenoptera, because they breathe through diffusion. If they get suffocated or melanized by the immune system of the host, they are screwed. So they have to be very in tune with their host to either dismantle or overcome the immune system. So then I think flies were able to acquire a greater diversity of hosts. And I'll keep you posted once I learn if that's true or not. I have ideas, I don't have practice yet. But this research has 3 goals. 1, to understand the ancestral life histories of parasitoid lineages. I told you that they evolved this about 20 times. How? From where? What was the most common pathway to parasitoidy in flies? Some studies suggest it's saprophagy, although nobody has tried testing this hypothesis. I also want to understand, it always makes this, the little thing that you can't read is frequency of parasitoidy. I want to investigate these respiratory innovations of parasitoid flies and how they change lineage diversification and life history evolution. If you have a siphon, do you diversify more? Do you have a greater diversification rate if you are an endoparasitoid? The way you breathe, does it affect macroevolutionary patterns? And then I want to see how all that ties into the hosts they use. I am fascinated about these relationships where there's a lot at stake for one and the other, and they're tight and they're under strong selection. Oh my God, I fixed it this morning, adaptations and adaptations. That's what he said there. I want to understand the ecological behavior and phenotypic adaptations of parasitoid flies and how they affect specialization in host switches and all. And there are a lot of students in the lab that either are still currently in the lab working or elsewhere still working on these projects on all kinds of groups, from the whole flies themselves to the specific groups of flies. So we have bee flies, we have snail killing flies, and then the borderline between the other sides of parasitism too, such as kleptoparasitism, where they steal the food, not necessarily the bodily fluids, and interactions with other hosts, such as plants. There's lots of going on in terms of macroevolutionary analysis in flies in the lab. I also want to show you what we've been doing in terms of integrated specimen-based research. I do lots of research with morphology, do lots of research with the specimens themselves, but I care about them as sources of genetic data as well. And you all heard about museomics. It's all the rave right now, but it works. I mean, when it came out, I was like, yeah, sure, it works. You can get enough and good DNA data from museum specimens, meaning each individual specimen in the Lyman Museum or all collections around are valuable as sources not only of biological, phenotypic, temporal data, but genetics. So now we can revisit biodiversity research and harness the data in the collections genetically. And we now embarked on this thing. I got the funding. I'm waiting. Apparently there's supply chain problems, but soon we're gonna have the Lyman Lab of Insect Diversity and Phylogenomics at McGill. This is a $1,000,000 project to revamp the labs that we have here, build new, and acquire the fancy toys for us to be able to both generate genomic data in-house and store it for posterity. I got this giant -80 freezer for tissue samples, so where we can store the specimens themselves, that I want to make available and online for everybody to use, and the extracted DNA, plus the genetic data freely available for anybody to use. So these are the plans. So now we'll be getting the fancy ones where you can kind of see things. So maybe on the next talk, it'll be like the next step until hopefully in 2024, early 2024, I'm hoping this will be done and we'll be able to generate and store genomic data in the house. What to do with genomic data? I'm talking about genomics and all. Well, the genome is vast. There's a lot going on, and not all parts of the genome is reliable or useful. So my research also, I'm going to talk very briefly about this because this is a lot of statistics, and we're here for the fun part, not necessarily the nitty-gritty details. But basically, my research works on bioinformatics and genomics to understand how we can select the parts of the genome that we can model, because computers are a luxury sometimes, and if we all use whole genomes all the time, we might not be able to analyze it all, and we don't need to analyze it all. So how do we select the parts of the genome that we can model? Because this is all tied into a model. A model is a simplification of a complex reality, and if it was faithful to reality, it wouldn't be a model. It wouldn't be useful. We all know the statement: all models are wrong, but some are useful. Which ones are the useful ones for the data that I have right now? It's not about the models being wrong. It's about which ones are best for my data. And we do this through a statistical approach called posterior predictive simulation, where we use the real genetic data, simulate data sets, do lots of statistical comparisons, and we are able to assess how well that model fits our data. So before we run the analysis, before we plug it all in to analyze, we can figure out and sort out through all the genomic data which is the one that we can analyze, because we have much more data than we have models to analyze them. And we have much more data than we have computing resources to analyze it. So when the bottleneck is on computing resources these days, we need to be careful. And my professor for my PhD, one of my favorite ones, used to say artisanal data sets. And I like that. Let's come up with artisanal data sets, because, you know, someone that might not have access to computers might be able to analyze a smaller subset and get enough great results just with whole genomes, if not better, because if you have too much, you may add more bias to it. And another thing, and I think, let me see, I'm almost done. what I care a lot about is how to translate all that I talked. Those are specialized genetics, specialized statistics, specialized everything. How do we translate all that and trickle it back to communities? I don't like the term citizen science. I'm not a citizen in Canada. So if we're talking citizen science, I'm not included. So I like community science better. There may be better ways to reframing it, but citizen kind of has a connotation to it. I want to understand and I want to promote crowdsourcing, community engagement, and how we can share the love and knowledge of insects. And several students in my lab are very skilled, and those are all drawings from one of the students, Sarah. And we are developing these tools for outreach in the greater Montreal area. These are the first. We introduced the common insect orders, and then we are gonna do one for the garden pollinators, and then the most common insects in the summer, and the common wasps, and why you don't have a murder hornet, why this little beetle is not gonna destroy your life. So that kind of thing, because I get those emails all the time. So I really care. Spiders, Catherine is saying, yes, yes. But Catherine, you don't need me. You do such a wonderful job and you do so much better. If anything, I need to mirror what you do for spiders and then plug it in on insects. You don't need me. So last thing that I want to show, and this has some connection to specimen-based research and genetics and everything, but it's a project that's more applied in agricultural than anything. It's to understand how insect phylogenetic diversity translates to pollination services and how we can promote biodiversity in cranberry farms. So we want to understand how pollinators communities respond to different practices, especially for cranberries, which mean a lot in Quebec. They're very, very a large industry. And this project is done with your very own Dr. Andrew Young. It's in collaboration with Andrew. And we got the funding. We did the first round of collecting this year. I owe an email update to Andrew, and I mess. I mean, I know. I'm sorry. So I want to just check it here because this is new to him too. That's how bad I've been with communicating. So we want to see how pollinator communities are affected by different things that the farmers do, and how these changes into the pollinator communities affect pollination services by managed and wild pollinators. And this knowledge can be used to circle back to best practices for growers. This is how you promote free pollination by wild pollinators in your cranberries. And these are the things that promote biodiversity. And it can also provide, we're hoping, provide some evidence-based information for policy as well as outreach. Everybody cares about sustainability and everybody cares about biodiversity. Why not link the two things together and use it in terms of insects and specimens and phylogenetic resources? And that's it. And I really hope I didn't go over time. I did 1 minute over time. Oh my goodness. I guess I'm gonna be talking with you over emails.
Well, you know what? I think we've got, we've just all experienced 900 odd days where time can be extremely relative. So I want to take this bit of existential time and encourage you to reach for your emojis or your camera to turn on and join me in thanking Dr. Gillung for a super inspiring and enthusiastic invocation to the importance of specimens and samples and curiosity and equitability and. So I'm not going to change the start time for the posters. That's going to be 1:45, but I would say let's do some questions right now, because I would hate to let me also just stop the recording there.
I am so sorry I went over time.